Hi, my name is Hoyan Lai. The title of this poster is AI Microscopy. Deep learning minimizes the impacts of fundamental microscopy limitations. For light microscopy, there are three limiting factors to consider that are shown in Figure 1 to the right of the introduction: light exposure, temporal resolution, and spatial resolution. There are trade-offs and compromises to be made between each of these factors to get optimal balance in terms of image quality and sample viability. We can reduce the impact of these trade-offs with deep learning. That's been shown with the CARE paper and many others. Here, we will apply a network architecture called 3D Arcan to extend live cell imaging, to reduce noise, and to enhance the image resolution. A diagram of the 3D Arcan architecture is shown in Figure 2 on the left. The network uses a residual and residual structure. Each group in dark blue is composed of several residual groups in light blue. The light blue groups is made up of residual channel attention blocks, and there are short and long skip connections between each residual group. These skips bypasses the low frequency information so that higher frequency information can be learned. And the result is that we can restore low SNR image to high SNR using this model. Learning the high frequency information is crucial because that typically contains the important structural information on the image. We use the 3D Arcan network to train a model to remove the image noise. We trained the model using pairs of images acquired at low light intensity as input and high intensity that's been deconvolved as ground truth. The 3D Arcan restoration results are shown in Figure 3. This is an image of Emerald Tom20 labeled mitochondria in fixed U2OS cells that's acquired on an instant SIM system at 60x magnification. We also compared our networks against a couple of other networks, particularly CARE, SRSNet, and ESRGAN. In 3A, we show the output in XY and an axial reslice in XZ. Visually, you can see that the Arcan looks to be most similar to the ground truth, both in XY and XZ. This also bears out in quantitative analysis in 3B, where we looked at the 3D structural similarity index and the peak SNR. The Arcan performs all the other three networks with CARE in second place. With this model, we can also restore images in live cell imaging. We use a much lower light intensity to image live U2OS cells expressing P shooter GFP in the mitochondria and M-apple LAMP1 in the lysosome. We required an image volume every 5.6 seconds over 4 hours, and that comes out to be about 2,600 volumes in total, and restored the data using our trained model. This allows us to observe the interactions between the mitochondria and lysosome in real time and count the number of different events, such as fission, fusion, and contact, without sacrificing the health of the cell. And you can see that in the video that I'm going to show you right now. Since the original Arcan paper describes a method for image super resolution, we turn our attention to using our network to enhance the quality of confocal images, which you can see on the right half of this poster. We required pairs of confocal and set images of MEF cells in dual stained for microtubules and nuclear pore complexes, which are shown in Figure 5A and B. The restored image resolution matches that of the set data both quantitatively and quantitatively. We also tried training the model to improve resolution of live confocal nuclei images, which you can see in Figure 6 and 7. A challenge with this data is that the chromatin takes on a very different appearance during the division, and that can throw off the model. We gave the model input data and contains the nuclei during the different phases of the cell cycle in Figure 6, and then applied the model to live cell data acquired in resident confocal mode in Figure 7 on the right. The chromatin structure are greatly improved by this model. To summarize, we use a deep learning network called 3D Arcan to try to overcome the inherent limits of light microscopy. We show that the network performs well for reducing image noise on ISIM images and for improving confocal image resolution to a level comparable to STED. The models can be applied to live cell imaging that allow us to extend imaging duration and to see the chromatin structures better. We are continuing to validate the network and quantify the errors. The models and code can be found in our repository and on our website. You can read more about the 3D Arcan architecture and some of the additional results that we are not reporting today on our paper that is currently in preprint in bioRxiv. I want to thank Gigi and Hideki, who are the lead authors on this paper, Jiamin and Yichang for performing the validation on 3D Arcan and the other models, Yijun, Alex, and Chris for acquiring the images, Hung Yu and Chi Chao for maintaining the GitHub repository, and James Lee, Luciano Lucas, and Hari Shroff, who are the principal investigators of this project. Thank you for listening.